I'll just get started right away. So I'm a postdoc at the Broad Institute and the MIT Center for Statistics, and I'm excited to tell you about analyzing developmental processes with optimal transport. So we're motivated by Waddington's epigenetic landscape, which is a classical metaphor for how cells develop as an embryo grows, and even how your cells even destabilize with age. So there's kind of three levels of easy, medium, and hard questions that we might like to answer about processes like this. So the most basic is, what cell types are there at any given stage of development? And you can answer these with basic questions like clustering, for example, okay? And the basic idea of this valley is that each of these little valleys would correspond to a different cell type that you might have at the end of a developmental process, where the process might start at the top with something like an egg cell or a stem cell, and then over time, the cell would roll down like a marble through these ridged troughs into different terminal cell types. Okay? So the medium question that we'll focus on today is what trajectories might cells follow? And we'll hint at the very end of the methodology by addressing what regulatory programs shape the landscape. Okay? So we're motivated by this technology, single-cell RNA sequencing, which I'm sure many of you are familiar with, which can now, with the dramatically reduced cost of sequencing, we can now record hundreds of thousands, or some people record millions of expression profiles of individual cells. And we can do this over time to record the locations of cells as they roll through this landscape. But because single-cell RNA sequencing kills the cell, like in the basic first step, you lyse the cell and you amplify the RNA and then you sequence it as cDNA. Because you kill the cell, you can't actually measure an individual cell at multiple points. But what you can do is record samples from this process at different times. And by recording these snapshots, our goal is to reconstruct the shape of this landscape. So that's the overarching goal, is to recover these trajectories from single-cell RNA sequencing time series. Yeah?
What would the y-axis be in that graph?
The y-axis is just like a metaphorical potential or something like that. But we won't even, we're going to abstract it a little bit. So we're going to think about an abstract stochastic process, okay? So here, there's actually a y-axis in this graph is actually 20,000 dimensional gene expression space, okay? And this is one-dimensional time on the x-axis. So it's 20,000 dimensions because there's 20,000 genes, right? All your cells have the same DNA, but they use it in different ways by expressing different genes. And there's a dimension for each gene, and the value of the cell along that coordinate is the number of molecules of mRNA. Okay? And then we measure this with noise because we recover a limited amount of mRNA from the cell, etc. But we wrap all of that noise, both the biological kind of distribution overexpression and the measurement noise, into one distribution that's changing over time. And this is this stochastic process on gene expression space. Okay? That's why I use the word developmental process, because it's suggestive of this stochastic process. And yeah, we assume it's Markov and stuff like that. But okay? So the coupling of a stochastic process describes how the distribution changes over time, right? So this is precisely the information that's lost by single-cell RNA sequencing, because we kill the cell. So mathematically, a coupling is a joint distribution on pairs of time points. So if you want to think about the conditional distribution, you might ask, given that you're a cell here at time t4, where would you likely be at time t5? And the transition matrix for the process, it's not really a matrix because it's a continuous object, but the temporal coupling, which you can think of like the transition matrix for a finite dimensional Markov chain, describes these conditional distributions and joint distributions. Okay, so now we can formalize our goal mathematically as recovering this coupling from independent measurements. So we write here p hat. So our original stochastic process was p. P hat is the empirical distribution, which is formed by taking all of the samples at a time point and putting delta functions on each one. So this is the empirical distribution, the ordinary thing in statistics. Okay? And we want to connect these empirical distributions with a coupling, right? Because what we observe is just the empirical distribution, and they're independent at different time points. Okay? So, yeah?
Maybe you will get that, but if your time points get dense enough, I think at some point within the time point, maybe you will also already capture different also time points and
Yeah, so the distributions here might overlap a little bit, you're saying?
Yeah.
Yeah, that's totally possible. Yeah, so that would be something like asynchronicity in development. And that's possible, but this PT just encodes all of the cells that exist at a particular time point. And
Is that real time point, or is it kind of developmental time?
For us, it's a real time point. So we're thinking that,
For a certain amount of time, and you measure all
right,
the cells.
exactly. So we imagine that at some real time, the cell, or the collection of cells has some distribution on gene expression space. And what the experiment measures directly is this entire population of cells gives rise to this entire population of cells at the next time point. And what we'd like to infer is that this subpopulation gives rise to this subpopulation. We don't measure that because we don't know that these cells give rise to these. We just know that as an entire group, this entire distribution at real time t1 gives rise to the entire distribution at real time t2. So our approach to estimate this temporal coupling is called optimal transport, and this is a classical field in mathematical analysis. It was initiated by Gaspard Monge, who's the father of differential geometry, and he invented the change of variable formula in calculus. And so this is one of his pictures here of how he planned to move dirt from one position to another. So he was building fortifications, and he worked with Napoleon. Apparently he was one of Napoleon's closest friends, and they went to Egypt together and built a lot of fortifications and did, I think he mostly did ballistics calculations, but this was one of the new mathematical things that he did.
Do you have to make an assumption that this coupling is invariant across time?
No.
I suppose that coupling will change across different time points.
Yeah, between each pair of time points, there's a coupling. Yeah. So there's actually, for any pair of time points, there's a coupling. And when we get to the analysis section later, we'll see that over short time scales, we believe it's like optimal transport. So just to kind of give intuition for what optimal transport is, you can imagine that if you have this distribution at time t1, the red one, and you want to transform it to another distribution, so for Monge, this was a pile of dirt. Maybe this is like a mountain that's tall here and flat here, and you want to build a castle on top because the British are coming. So then you have to flatten out the foundation, okay? So you have to transport. So the least efficient thing possible would be to take, this dirt over here and lift up all the dirt to this ground level here and move all this dirt to this side, right? And move all this dirt to the, move the right dirt to the left side and vice versa. So that's extremely inefficient. So you can kind of intuitively imagine that it's more efficient to, move this dirt here and move this dirt here, right? But in order to specify how to do this, because there's a lot of different ways to do this, you need to come up with a plan called a transport plan. And that basically minimizes the total work, or total mass times distance that all of the dirt moves. And we'll see in a minute that this is a linear program, and it was rediscovered by Kantorovich in the 1940s and used to transport goods from factories. So it's used today in a lot of different areas. It's used in computer graphics, and they obviously have the nicest pictures. So this is a continuous interpolation between a cow, a duck, and a coffee cup. So it's used in pure math and differential geometry. In statistics, you can use optimal transport to compare probability distributions. Like, for example, you can use the KL divergence to compare probability distributions. But here we have empirical distributions. So the KL divergence and total variation don't work well with empirical distributions, or for any distributions that have different support. It's used in economics, like I said, to transport goods and stuff like that efficiently. And it's also used in fluid mechanics to analyze flows of incompressible fluids. Anyway, so yeah, in order to figure out how to transport the dirt, we need to come up with a transport plan or transport map. So mathematically, what this tells you is that if you pick up a column of sand at position x, okay, so we think of this as a delta function at x, then the image under the map is described by some distribution. So you don't have to put all this dirt down in the same place. You can spread it out a bit, okay? So the image is this pi of x. It's a little pile, pi. And the optimal transport plan minimizes the expected cost. So if it costs c of xy, some cost function c of xy to move a unit mass from x to y, and pi of xy describes how much mass we move from x to y, then the total cost is given by integrating over c d pi. Okay, and it's a linear program because the constraints we have are these two linear constraints. So these are linear in pi. Okay, so the constraints are here, you can easily interpret these as, this first one says that if you sum up over all x where the dirt came from, then you should get the total distribution at time t2 that you ended up with. And vice versa, if you sum up over everywhere the dirt went from all x's, then you should get the dirt coming out of position x. So it has conservation of mass fundamentally built in, just fundamentally built into the setup. Okay? Now, given finitely many cells, these integrals are all sums, okay? And the optimization variable is a matrix. So when we're transporting the blue cells to the green cells from time t2 to t3, we would solve for a transport map, which would be a matrix where there would be a row for every cell at time t2 and a column for every cell at time t3. Okay? And for example, if you consider this green cell at time t3 and you look at the transport map on the, if you look at this transport map, then this specific column will tell you the distribution of the ancestors. So where did the dirt come from? Or where did the mass come from for that cell? And maybe it's easier to think about going forward in time. So if you consider again this green cell here, there's a row for the transport map going forward in time, and this tells you the distribution of where its mass goes. So I've used color here to try to indicate maybe the mass of the distribution is big here, so it would be big here and small here. Okay? So we have a linear program with a matrix valued optimization variable, and
You need to specify that cost function.
yes, you do. We use Euclidean distance, like squared Euclidean distance. The basic example, like the basic way to think about cost functions is like in economics, sometimes you want to use a concave cost function because it's more efficient to put all of your loaves of bread on one truck and ship it all the way across the country. Whereas in if you have a bunch of books on a bookshelf and you want to move them all one to the right, and you use the linear cost function, then it's the same cost if you move every book one to the right, or if you move the one book all the way to the end. But if you use, then in the other extreme, if you use a convex cost function, like Euclidean distance squared, as opposed to square root of Euclidean distance, then if you use Euclidean distance squared, then it's more efficient to move all the books one to the right, and that's the cost function we use. Because we don't think it's reasonable that one small group of cells moves a huge distance and explains everything. Anyway, yeah, another question?
Yeah, so optimal transport linear, but probability density estimation is not linear, it's like 20,000 direction. So what do you do?
We use empirical distributions. So we transport the empirical distribution to the empirical distribution at the next time point.
What do you mean by empirical?
So an empirical distribution is a sum of Dirac deltas on the observed data. So we don't do density. We don't do any density estimation. So we don't put a kernel on each observed data point and like estimate a smooth density. We just put a delta function. So we don't we're not transporting densities that are continuous with respect to Lebesgue measure or anything. Yeah.
Discrete density.
It's not a density. Yeah, it's
That's like a histogram? Oh, yeah, it is some sort of density.
it's just we're transporting the observed cells at one time point to the observed cells at the next time point. And the transport map says nothing about a new cell that you.
Just mapping what cell goes to what cell in the next one.
Yeah, right. It's like a matching.
Oh, it's like a matching.
Yeah, yeah.
That was going to be my point, is I feel like you could think about it without thinking about a distribution at all, right? You could think about just you're trying to do matching, but it's like soft matching rather than hard
Right.
matching like one cell to another.
Yeah, so if you wanted to do, using a transport map is a relaxation of it's a convex relaxation of the matching problem. That's right. Okay, so the basic idea is to match the cells at one time point to the cells at the other time point with the transport maps. But like I described, there's conservation of mass built in. And cells can proliferate, but dirt doesn't proliferate. So we have to change something. We have to change this conservation of mass. And the way that we do that is we introduce a growth function, g of x. So if we pick up a column of dirt at position x, and then it's actually a pile of cells and it grows at rate g of x for some time, then when we put this pile down, it could have more or less mass. Okay, so this is like a big pile, and this is a small pile, so with big or small growth rates. And assuming we know this function g of x, then we can put it into the constraints in the following way. So now, if you sum up over everywhere the dirt went, all the y's it went from some position x, then it should start off with the starting mass times, then it gains this additional mass factor. Okay, yeah?
So again, this is a problem because you don't density estimate, right? If you have two density estimates, there's no mass conservation problem. You only had it because you're counting cells in this.
So we can have a different number of cells at 2 time points, is that what you're saying?
So that's right, but instead it's you estimated density here and density here, and then you use optimal transport. How it's supposed to be used into your problem.
It's a problem if you have heterogeneous subpopulations. So for us, this distribution is an empirical distribution. So if we have n cells at one time point and n2 cells at the other time point, then we divide.
Density estimated doesn't matter, right? Probability density is added to 1.
Yeah, that's right. They do add up to 1. But if you have a
Yeah, normalization.
right, but there's another problem. So imagine that you have 2 subpopulations, okay? And at one time point, so we normalize them. So at this time point and at this time point, they both have mass 1. But if it's like 50-50 here, but this cell type is dying, so here it's 30-70, then what optimal transport would do, it wouldn't know that these can die, and it would just transport these here instead. So that's what we're addressing with the growth rate. Okay? So yeah, you're totally right that we can normalize each of these, but there can also be non-uniform growth across the cells at any time point. And if you don't know that these cells give less mass to the next generation, then you would erroneously see this transport from this subpopulation to this subpopulation. Does that make sense?
What is the direction of transport?
So this is time. And this is, sorry, yeah.
Because in this slide.
Oh, I'm sorry. Okay, well, in this picture, it's different. So, but is it clear what I'm trying to explain? Where like if there's 2 populations that have different,
So
no, so it's a convex optimization problem if you assume you know G exactly, right? But we don't. So what we do, thank you, is we we relax this equality constraint, okay? And so we penalize the KL divergence between this distribution and this distribution, okay? And so now this is a convex optimization program that's no longer linear, but it doesn't need to be a linear program. So we don't know G exactly, but we have an initial guess, and we allow for some mismatch up to some tolerance. And we also, one more thing we do is we add, people in the transport field know that it's a good idea to add an entropic penalty. So what this does is it promotes entropy in the transported mass. So as opposed to a perfect matching, you get cells going to different places. So there's kind of three reasons that we might want to have entropy. Firstly, biologically, to what extent are cells fated? If we just have a perfect matching with no entropy, then our estimate is going to just say this cell goes to this cell, right? But maybe a cell might have two possible ways it could go. So there might be some biological component to the entropy that a cell's fate is not fully specified. Secondly, there's a statistical component to the entropy, which is that we have finitely many observations, and they're noisy observations. And so if some cell in my rotated time picture was started off here, and it had some true descendant, say it only had one true descendant at this time, then if instead we only measured a few cells in that region, we wouldn't actually measure where it truly went. So, we'd want to put some mass in that neighborhood, to avoid overfitting, to say we're 100% confident it goes to this position. The third reason that we might include entropy in transport is purely computational, just because it makes the objective function strongly convex and we can solve it like way, way faster. Okay? Yeah.
So is it more like a sinkhorn with?
It's sinkhorn, exactly. Yeah.
With some moment constraint.
Let's see. The moment constraint.
The difference between
It's
the sinkhorn is the KL that you have to
so right. So Marco Chiuturi had a paper in 2014 showing that you can do sinkhorn iterations for optimal transport in the classical, in this case, but with entropy, but with equality constraints, But now when we have these unbalanced constraints, there's a new paper from 2016 showing you can still do the sinkhorn iterations. I can talk about it offline with you.
Quick question. Do you know the extent to which the regularization shifts you off of the true optimum? Does your program still recover the correct optimum?
What do you mean by true optimum?
Well, because if you apply shrinkage regularization, for example, then it warps the coefficients at the end.
Yeah, there's bias.
Yeah, it will introduce some bias. So are you able to eventually correct for these bias?
Well, we don't want to correct. We don't think, because there is some biological component to the entropy, so we don't think this zero entropy object is even the right thing.
I see, okay.
Yeah. But intuitively, you want to add entropy until you don't want to start blurring out these transitions completely, right? So too much entropy will just give you the independent coupling. So here's another way, because we have some questions about entropy. So another way to think about entropy is in terms of something called a Schrodinger Bridge. So as you all know, a Brownian motion, if you start off Brownian motion at the origin at time 0, then at time t, you'll have a Gaussian with variance, with some variance that depends on t, right? So now a Brownian bridge, as many of you all know, is if you start off with a Brownian motion at the origin, and then you condition on the fact that it ends up somewhere, the Brownian bridge is the distribution on paths between these two positions. So a Schrodinger bridge is if you have a collection of indistinguishable particles. So there's 4 pictures here, so just think about this one for now. So if you have 1, 2, 3, 4, 5, 6 red particles at time 0, and then you have 6 blue particles at time 1, okay? And if all 6 of these indistinguishable particles are doing Brownian motion, then you don't necessarily know which particle went where because they're indistinguishable, okay? So depending on the diffusion coefficient of the Brownian motion, you might be more or less confused about which particle went where. Okay, so here I'm showing Brownian motions with different diffusion coefficients, and I'm conditioning on the event that they start off in the red positions and end up in the blue positions. So Schrodinger proved in 1932 that entropically regularized optimal transport, where the regularization parameter is equal to the diffusion coefficient of the Brownian motion will give you the maximum likelihood coupling of which particle went where. Okay? So the coupling tells you which particle went where. And then once you have the coupling, then it's a Brownian bridge. So conditional on the coupling, you have Brownian bridges for the paths. So that's another interpretation of entropically regularized optimal transport.
Was he using that language, not Singlet?
He was using slightly different language, and he wasn't using the word optimal transport. He was using, yeah.
Sorry, can you have that?
Yeah.
So, how do you know how much to regularize? There's a maximum likelihood there's only one.
Just conceptually, if you knew the Brownian motion, variance parameter,
I see, that would tell you
and you set that equal to the entropy regularization parameter, then that would be maximum likelihood. Okay, so now just to summarize conceptually what's going on, you can think of this time-varying distribution as like a, some like hidden Markov model where there's transitions according to some true coupling, okay, and let's call the true coupling gamma. Then there's the optimal transport coupling between any pair of time points, okay, and we're kind of imagining that these are close. So there's the true coupling and the optimal transport coupling. Now, so then what these distributions emit are samples. So we have a hidden Markov model that's emitting samples, so emitting empirical distributions. And what we compute are the transport maps on those empirical distributions. Okay? And it's well understood that these empirical transport maps concentrate around these population level transport maps. And this is the part that we'd like to analyze next. Okay? So this is kind of work in progress. But you can imagine that in the space of distributions, if we have some distribution that's changing over time, then this assumption that the true coupling agrees with optimal transport is kind of like saying it's a straight line over that time interval. So this assumption is not really reasonable over long time intervals because you'd cut corners intuitively. But we'd like to prove, and we have some initial work on this, we'd like to prove that the true coupling between any pair of time points can be obtained by composing a bunch of little tiny transport couplings. And the statistical version of this would involve finite data and stuff like that. So this type of theorem is proven in one special case by Felix Otto and others in 2002. So if there's a potential function phi, and this goes back to your question from the very beginning of the talk, what's the height of that picture? So if you think of the height of the Waddington landscape as a potential, okay, then if you imagine that the balls are rolling according to the gradient of the potential here, plus maybe some Brownian motion, so if they're rolling according to the gradient of the potential, then in this restricted setting, the true coupling is the composition of a bunch of infinitesimal transport couplings, if you had data at all those time points and stuff like that. But this is a kind of restrictive setting to think about potentials because this kind of says there's a slope, like a ski slope, and each cell is skiing independently of the other cells. But in reality, there might be something like cell-cell interactions, and our use of optimal transport, which goes beyond just this idea of a potential, we believe it can also include stuff like cell-cell interactions. Because the distribution that's changing over time, it's totally flexible and can incorporate cell-cell interactions. Okay. So how do we leverage this now to learn regulatory models? Okay. So with the transport maps, what we can do is we can try to infer trajectories of cells moving over time, because given a cell's at some position, we have an estimate of where it will be at some future time point. And now that you have these kind of pseudo time series, we can try to learn something like a vector field that describes this flow. So we see the distribution changing over time, and we now know which piece goes where, but we want to estimate what's making it go that way. So motivated by the stuff from the previous slide, we're going to try to learn a differential equation or something in discrete time that can describe the flow. So given that you're at some expression profile x at time t1, we have some idea of where you might be at the next time point because that's what the transport map tells us. And so we'll try to fit this difference with some function f. Okay? So we can do that by sampling training pairs from the transport map, because the transport map defines a joint distribution on pairs of points. So we can sample pairs xt1, xt2 from this joint distribution, and then we can set up a regression to minimize over functions f. Now this is the loss. This is the loss, so it's expectation over pi. But you can just think of sampling pairs and doing least squares. Okay, so we've tried very basic stuff with f, but you can imagine, like we've just tried, linear functions f that are low rank with sparse components, and this has led to some, nice descriptions of the data. Yeah, question?
It's a similar question to someone had earlier, but at this step, are you assuming this f is going to be the same across time points, or is it going to be different?
So in our initial version, yes, we did. So this f was the same, but you could solve for a separate f at each time point. So conceptually, there's no reason you have to. It's just we solved for like a rank 10 linear function f just because we wanted to use as much data as possible to try to learn it. But in principle, you could have a different f for each pair of time points and parameterize it with a neural network or something, and just go crazy. So
I have one more question, which is there some circularity here in that the optimal transport stuff is trying to make these pairs of x's that you would sample be similar, because they're somewhat going to be close, right? Because of regular
right.
Euclidean distance, and then you're learning this function is trying to map. So it's already picking pairs.
So yeah, let me explain why the transport is a nice initialization. So in this rotated time picture where time is going up, if you just look at random pairs, you might have like, a pair x1 here and a pair and like a point x2 here, and this would be a spurious transition. So you wouldn't want to train your f to try to fit that type of transition, right? So as a first step, we learn that we're only going to sample pairs of points where x1 is here and x2 is here. And then we would fit it to just this type of, and so we believe this is like a nice pre-processing step to learn this f.
I guess to me it feels like it should be a joint optimization problem. I realize that might be like completely infeasible, but like if you knew what this f was, then
yeah. So that's a very good point, because if you look at this, okay, so look at this equation, okay, and now imagine that I ignore this term and I minimize over pi, okay, subject to the constraints. This is the optimal transport objective function with a cost function that depends on f, okay? So there's a very natural alternating minimization step, so it's good intuition. So we just did one step of this in our initial test on this data, but in principle, you could try to learn the coupling, assuming there's no flow, because it's kind of like maximum likelihood for Brownian motion, and then you could try to learn a flow, right? And then, as a next step, you could then try to account for that flow, like everyone's flowing, everyone, if you're at the airport, you're standing on one of those things, everyone's going along the same conveyor belt, right? Then given that flow, what's the Brownian motion on top of that with this cost function? Okay? So I think how much time do I have left?
I think we started a bit late, right?
Maybe 5 more minutes.
5 minutes, yeah. So that's essentially all I wanted to say about the method, and in the last 5 minutes I can just show pictures about the experiment. So, okay, so we tried this on reprogramming fibroblasts to induced pluripotent stem cells. Okay, so we took time points over a 16-day process, and we made independent time points by generating a plate of reprogramming cells, and then we sampled an entire plate to make a time point. So these are independent measurements, like our setup. And we made a movie showing, here each point is a cell with 20,000, genes, and we embedded it in 2 dimensions. And these are the points kind of moving over time. And this is kind of the final time point. And you can see that here's the previous time point, next time point. So going back and forth, you can see that the orange cells in this branch here all kind of largely disappear. So we think these cells are dying. And the cells on the top are successfully reprogramming. And we can use the transport maps to identify trajectories. So we believe these cells that successfully turn into IPS cells come, in this path, and we can just take the transport map and, propagate this distribution back in time, starting from the uniform distribution on these red cells, which is just a subpopulation of the final time point. We can propagate it back in time, and we can also look forward in time, and we can see that there's some early time point. So at day 2, we can differentiate between the fates here already. So this black spike is giving rise to these cells more than it is to these blue cells. And so this group of cells will be the ones that eventually reprogram, etc. We can compare our prediction to a previous experiment, and we agree for many of these cases. So these are all different cell populations. So cells that are positive for CD44, some marker, negative for some other markers. These cells have some percent chance, like 22%, of being IPS cells at the end. And people measured that empirically, and then we estimate that from the transport maps. And for all these different options for these markers, except for this one for some reason, we kind of agree. So this is some very, very, very basic validation of the transport maps. And the final thing is we can infer regulatory programs by solving this low-rank regularized regression that I was talking about. And we identify a set of transcription factors that are pushing you in the direction of pluripotency, pushing you towards being a stem cell. So this is, here I'm showing the loadings on one of the rank 1 components. And the top 2, nanog and this other one are both known genes. This one, number 3, OBOC6 was a gene that wasn't known to be related before to reprogramming, and we found that it doubled the chance of cells becoming IPS. So we ended up with twice as many IPS cells when we turned that gene on artificially at the end. Yeah, so that's it. That's the conclusion. So just to wrap up. Next steps that I imagine are measuring the growth rates experimentally, and you can also imagine optimizing those. Validating this OT principle. So we're currently doing this. We have a new data set where we have very dense sampling, so we have like 4 times as many time points, and we're going to try interpolating the distribution. Just like the computer graphics guys can interpolate between the cow and the duck, we can interpolate between distributions of cells and then see right in the middle, does it look like a cow duck, so it actually looks a little bit like a chicken in the end. Yeah, so that's it. And I'm currently working to analyze the theoretical properties. So thanks for your attention. Thank you.